Hello, welcome back to my YouTube channel, and in this video we are going to look at the effects of insulin on glycolysis. If you haven't watched my other videos, please go back and check them because that's going to be the foundation for you to fully appreciate this video. So in the previous videos, we talked about the GLUT2 and the GLUT4 transporters. We said that the GLUT2 transporters are found in the beta cells as well as the liver cells, while the GLUT4 transporters they are found in the muscle cells and the fat cells. And what you should know is that the GLUT4 transporters, these are insulin dependent. They are the only GLUT transporters that are insulin dependent. But the GLUT2 transporters, GLUT1, GLUT3, GLUT5, and others, they are not insulin dependent. So what do we mean? We mean that GLUT4 transporters, they are found inside the cell, in the cytoplasm. So they depend on insulin for them to move and go and bind to the cell membrane. But all these other GLUT transporters, they're already embedded in the cell membrane, so they don't need insulin at all. Now, let us imagine that you have got a muscle cell. So in a muscle cell, glucose is going to enter via the GLUT4 transporters. When it enters into the muscle cell, this glucose is going to be phosphorylated. Why are we phosphorylating glucose? Because we don't want glucose to leave the cell. Why? Because the GLUT4 transporters and other GLUT transporters, they are bidirectional. What do I mean bidirectional? I mean that glucose can enter through these channel proteins and it can also leave. So glucose can enter through the GLUT4 transporter into the cell and it can leave the cell through the same GLUT4 transporters. So for us to trap that glucose inside the cell, we have to add a phosphate, as you can see here. So glucose is going to be phosphorylated to glucose 6-phosphate. Now, we have got two types of enzymes that can do this. We have got hexokinase and glucokinase. And there is a significant difference between these two enzymes. Now, what is the difference between hexokinase and glucokinase? So the main difference is that when we talk about glucokinase, this enzyme, it is only found in the beta cell and the liver cell. Hexokinase enzyme is found in all other cells apart from the beta cell and the liver cells. The other thing that you should know about these enzymes is that hexokinase, it is not insulin sensitive. So when we talk about hexokinase, it is not controlled by insulin. Glucokinase is controlled by insulin. Glucokinase is found in the liver and the beta cells. Hexokinase is found in all other cells apart from those two. Now, how is glucokinase controlled by insulin? Remember that in the previous video, we talked about the AKT or protein kinase B. Protein kinase B was activated due to the presence of insulin. So AKT, that was an enzyme that was phosphorylating different reactions. So even here, AKT or protein kinase B, this enzyme is going to come. So I've got protein kinase B right there. This protein kinase B, what does it do? This enzyme is the one that phosphorylates glucokinase. So if it's in the liver, imagine this process is taking place in the liver. If it's in the liver, we've got glucokinase. And this enzyme, it is activated when it is phosphorylated or when AKT, also known as protein kinase B, adds phosphate group to glucokinase. So glucokinase is going to become active. And when it becomes active, it will start converting glucose to glucose 6-phosphate. But if it's in the muscles, we have got hexokinase, which is not controlled by AKT. So if it's in the liver, AKT will activate glucokinase by phosphorylating it. But if it's in the muscles, this AKT has no effect on hexokinase. That's what you should understand. The other thing that you should understand is that glucose 6-phosphate is converted to fructose 6-phosphate. This fructose 6-phosphate, it can be converted into two different kind of molecules. Now, what are these molecules? The first one is fructose 6-phosphate can be converted to fructose 2,6-bisphosphate by an enzyme which is called PFK2, phosphofructokinase 2. Now, this enzyme, what you should know is that it is activated by AKT, also known as protein kinase B. So, protein kinase B, it phosphorylates PFK2. And when PFK2 is phosphorylated, this enzyme will convert fructose 6-phosphate to fructose 2,6-bisphosphate. But why are we making fructose 2,6-bisphosphate? Remember, fructose 6-phosphate can also be converted to fructose 1,6-bisphosphate. Pay particular attention to these two different kind of molecules. Fructose 1,6-bisphosphate and fructose 2,6-bisphosphate. These are different kind of molecules. There is two, there is one. So fructose 6-phosphate can also be converted to fructose 1,6 base phosphate by an enzyme which is called PFK1. But this enzyme, it cannot work without fructose 2,6 base phosphate. What do I mean? Fructose 2,6 base phosphate, please take note of that. Fructose 2,6 base phosphate, this molecule, it is an allosteric activator of PFK1, phosphofructokinase 1. So PFK1 depends on the presence of fructose 2,6 base phosphate. So when you produce fructose 2,6 base phosphate, this molecule is going to bind to the allosteric site of PFK1 and activate it. So PFK1 is going to be activated, then to start converting fructose 6-phosphate to fructose 1,6 base phosphate. So what is the overall effect of insulin on glycolysis? So what you should know is that insulin, it activates glucokinase and it also activates PFK1. How is it activating glucokinase? Remember, insulin was activating a protein or an enzyme that is called AKT, which is also known as protein kinase B. Protein kinase B, it phosphorylates glucokinase in the liver and the beta cells and activate this enzyme. This enzyme is going to phosphorylate glucose to glucose 6-phosphate. What else does insulin do? So insulin, through AKT or protein kinase B enzyme, it can also phosphorylate PFK2. So PFK2, this is a very important enzyme because this enzyme, it converts fructose 6-phosphate to fructose 2,6 base phosphate. And once we make fructose 2,6 base phosphate, so this is a very important allosteric activator of PFK1 enzyme. So fructose 2,6 base phosphate is going to bind to the allosteric site of PFK1 and activate it. PFK1, when it is activated, it will start converting fructose 6-phosphate to fructose 1,6 base phosphate. And glycolysis is going to progress. So what you should understand is that step 3 of glycolysis, it's a very critical step in this pathway. And what is the activator of PFK1? The main activator is fructose 2,6 base phosphate. And this is controlled by insulin through an enzyme that we call protein kinase B or AKT. So please make sure that you understand that point. So this is all I got for you on the effect of insulin on glycolysis.